Okay, so let's get started. So happy New Year, everybody. We're going to start a new year with a seminar series, and on behalf of Xinwei Wang and myself and the entire NCI CCR liver program, we are super excited to have Lopa Mishra speak today and give us their virtual seminar. So Lopa is a professor of medicine and Marinov Edward Chair, co-director of the Institute for Bioelectronic Medicine at the Feinstein Institute for Medical Research, Northwell Health, and Cold Spring Harbor Laboratory. She has been, working on different aspects of liver diseases and liver fibrosis and cirrhosis for a long, long time, and has focused on one of the important findings or studies has always been around TGF-beta, which disruption then leads to fatty liver and potentially cancers. And as part of their study, she actually also found that there is a human condition where the TGF-beta pathway is the effector pathway for human stem cell syndrome with an 800-fold risk for cancer. And when I read an 800-fold risk, I think, that's actually quite a significant number. She has had so many publications in more than 370, leading to more than 370 articles in the very, 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 very best journals, including Science, Cell, Cancer Cell, Gastroenterology, etc. And obviously, along with those papers, she also received many, many awards and is seen as speakers and at many, many conferences. She has received awards from the AGA. She's been awarded for the top women in gastroenterology in 2008. She was elected as a fellow of the American Association for Advancements of Science in 2016, and in 2017 she was elected to the governing board of ASLD, and she has been elected to become a member of the Association of American Physicians in 2020. And received an American Association for Cancer Research Team Science Award, I believe also in 2020. So with this, I would like to hand the gavel over so you can give the talk entitled Targeting Aldehyde-Driven Fatty Liver Disease and HCC.
Tim, thank you for the lovely introduction, and also special thanks to Xinwei Wang for inviting me to this very prestigious cancer seminar series. I'm honored, and I'd like to tell you a little bit about how the work we've been doing in targeting aldehyde-driven fatty liver disease and hepatocellular cancer. Now, can you all hear me? And also some of the work that we've been doing in biomarkers that capture that ongoing biology from mouse models to human disease onwards. Oh, so let's see. And like many of you, I see Sandy and Lola in the audience. We've gone from discovery and our mouse models and human genomics, the TCGA analyses, to identifying these right panels and treatment strategies. And hopefully sometime in the future, early detection and early sequential treatment with predictive evidence towards curing hepatocellular cancer. But as you know, hepatocellular cancer, it remains the third leading cause of cancer deaths worldwide, and it's rising alarmingly, and affects males 2 to 4 times more than females. This is something we still don't understand. It's rising with parallel to risk factors such as non-alcoholic steatohepatitis, now termed MASH, and alcohol-induced cirrhosis. So I bring this slide up. It's an old slide, but it shows that the demographics are changing. We used to consider viral hepatitis as a major cause for liver transplants, but that has now changed to alcohol-induced liver disease and MASH, particularly in women. So we could just stop, and you could say, well, we have to stop eating and stop drinking alcohol. But within ourselves, even as we speak, our cells are detoxifying formaldehyde and other aldehydes. And this has been uncovered more recently by aldehyde dehydrogenase 2, ALDH2, and the functional deficiency affects over 500 million people globally. So it's has a prominent role. It's an NAD-dependent mitochondrial enzyme, metabolizes reactive aldehydes that are generated as products of lipid metabolism, what we eat, and also converts acetaldehyde during the metabolism of alcohol. The mutations are dominant negative, and they destabilize the tetrameric enzyme. And in males, the mutations are strongly linked to significant metabolic differences. And they're associated with cancers of the liver, esophagus, and head and neck cancers. But then what about the pathways in hepatocellular cancer? You've all heard previous seminars where Wnt pathway, the Wnt signaling pathway, beta-catenin and MYC and p53 play a prominent role. But there's one significant pathway that is often overlooked because it's so complex, and that's TGF-beta signaling, plays a critical role in hepatocellular cancer. And this is a sort of tricky ground because it's a bit like taking coals to Newcastle. TGF-beta was discovered at NCI by Anita Roberts and Mike Spohn in 1981. And initially they thought it transformed and induced cancers, but it's been shown to suppress cancers. And of course, signaling was then discovered to occur through these globular SMADs, seen over here, that both suppress and can be harnessed for cancer. And TGF-beta then also signals through stress-activated kinases. But what I'd like to point out are two things. Almost 40% of human liver and gastrointestinal cancers have alterations in the TGF-beta members. And these mutations are heterozygous. So it's these haploid models which are really important, and dosage plays a massive role. If you look at SMAD3 knockouts and SMAD4 knockouts, it's these haploids that develop gastrointestinal cancers. But there are other molecules that really confer tissue specificity. And as we unfold the E3 ligases and adapters such as SARA, and one that we cloned, a SMAD3 adapter, Spectrin, that shows that they're critical for SMAD function. And it really came as a sort of surprise when we identified that partial deficiency in SMAD3 and spinding partners led to alcohol sensitivity in hepatocellular cancer. And this was dose dependent. And we noticed that there are high aldehyde dehydrogenase 2 levels in the SMAD3 mutants, as well as a Spectrin double heterozygotes. So perhaps aldehyde dehydrogenase 2 plays a protective role. And we weren't sure, and we hadn't figured that out. But suffice it to say, the Spectrin homozygotes are embryonic lethal. We published that in Science a long time ago. And then some mice go on to 11.5 and look like fetal alcohol syndrome, highly susceptible to alcohol susceptibility and toxicity. And the mice, the haploids, as I said, develop hepatocellular cancer. And as Tim very nicely pointed out, phenocopy of human cancer stem cell syndrome. So at the top, you see this alcohol sensitivity. You can give rodents a lot of alcohol, nothing happens to them. But the Spectrin, loss of Spectrin and SMAD3 really brings out this alcohol susceptibility. And these cancers develop spontaneously in C57 black 6 mice, but at a later age. So we wondered if aldehyde dehydrogenase 2 played a role, and could we then start building a robust cancer model by knocking out aldehyde dehydrogenase 2, and hopefully the mice would develop cancers, hepatocellular cancers, at 3 months.
Cancer treatments.
So we carried out an intercross between Spectrin and aldehyde dehydrogenase 2, and we obtained these mice from the Rockefeller Institute. And to our surprise, these male mice, seen over here on the right, at 3 months, are very fat. They develop severe and marked visceral obesity. The female mice do as well, but to a lesser extent. If you look on H&E labeling, the histology shows that there's macro and microvesicular fat, oil red O staining, as well as fibrosis. Well, so what does the Spectrin and aldehyde dehydrogenase do in human obesity and fatty liver disease. So we queried for polymorphisms in both of these in about 3,000 patients with human obesity and fatty liver disease. And we identified 52 SNPs, 7 belong to ALDH2 and 45 to the spectrum. Some of these are shown here. But then we focused on individuals with a high BMI, 30 and greater. And we identified 2 SNPs for the spectrum that really were significantly altered with a high BMI. So suggesting that these molecules have a human clinical significance and an association. Going back, these mice, as are seen on the far, in the left-hand picture, on the far right, are very fat. And the red bars show that the body weight is increased from 3 months onwards, and so is the liver weight. These mice develop cancers at a later stage, but not at 3 months. At 3 months, they're very fat, and later on, they develop hepatocellular cancers. So not to preclude that, but this story surprised us. Well, what about the spectrum? It seems like a stress integrating protein, integrator of stress. It interacts, has 2,000 interactions, and it represses telomerase with SMAD3 and a chromatin modeling protein, CTCF. And when you remove this repression, telomerase and IGF-2 are activated, and the mice phenocopy a human stem cell syndrome. But we and others have also shown that it's a substrate for caspase-3. It's a rod-shaped molecule, and at the 11th repeat, there's a caspase cleavage site. Now, if you eat a lot of fat, hepatocytes, normal cells would undergo apoptosis when caspases are activated, but hepatocytes undergo lipogenesis. And these spectrin cleavage products seem to be sort of co-opted pathologically into this lipogenic pathway. The cleaved products interact with the master regulator of lipogenesis, SRABP1, and we published that some 2 years ago, and stimulate de novo lipogenesis. So cleaved spectrin is harmful, which is really intriguing, because when you then stop these cleaved spectrin fragments from working, as Shuyun Rao, who was an assistant professor with me at GW, did, you find that hydrodynamic injections of targeting spectrin or blocking spectrin in the liver-specific knockout through albumin, or in a DEN, a chemically induced hepatocellular cancer model with the Western diet, or DEN alone, you can block both the fatty liver disease seen in the left panel at the bottom. You can block cancers, and together blocking these cleavage products can really, through this spectrum, can block cancers, suggesting that it could be a really powerful tool. But what about these reactive aldehydes that I've told you about? We found, which I haven't shown you, a lot of metabolomic and transcriptomic analyses, that these lipid esters were increased. And one molecule, one of these reactive aldehydes, is something that I shall focus on, hydroxynonenal. I mean, analyzed for expression of hydroxynonenal, it's an end product of lipid metabolism. And if you look at the top left-hand set of immunohistochemical labeling, in the spectrin and wild-type tissues, liver tissues, you don't see expression. However, in the double heterozygous, what we call the ASCO mutants, there's a marked increase in hydroxynonenal expression. So what is hydroxynonenal doing? And it's known to be involved in adduct formation, and through, similar to acetaldehyde, these form adducts and are toxic to cells. So we wondered if it accumulated, and adducts accumulated with hydroxynonenal, with treatment and without treatment. So the first plot shows you HUH and HepG2 and THLE, a primary human hepatocellular cell line, and this is just one representative picture. These cells on the left were not treated with hydroxynonenal. They're normal cells. There are no significant adducts. You add hydroxynonenal and you blot for HNE and you see all these adducts. When you block aldehyde dehydrogenase 2 with siALDH2, but you do not add HNE, you find a significant accumulation of HNE, 4-HNE adducts, suggesting that these adducts accumulate without without ALDH2, and this could be a significant pathological event in at least those 500 million people that we have ALDH2 deficiency. So what is hydroxynonenal doing? And we've identified that it interacts with the spectrin, full lead spectrin seen in this IP in immune precipitation assay, as well as the cleaved end-link part fragment of spectrin. And we went on to confirm that. But what we further went on to do was carry out alpha-fold analyses for molecular docking studies. And we found that Krishanu Bhowmik identified 2 sites that could interact and between spectrin, these are spectrin fragments, and 4-HNE, and form these covalently linked bonds, which we then went on with Paolo Cifani to determine by mass spec studies. And as you can see, there's a cysteine at 1284 that really interacts on mass spec studies by Michael and Schiff base additions. And so there's a covalent interaction and modification by this reactive aldehyde, which is cleaving the spectrin and then modifying it. And what exactly was the function? Well, I've already shown you that SMAD3 is a critical partner for the spectrin. So we queried, and our hypothesis was perhaps TGF-beta signaling is disrupted. And this is why we're seeing MASH and hepatocellular cancer. In human patients with MASH and MASH particularly, high TGF-beta levels are noted, and there is a disruption. So we treated cells, again, the same cell lines with small interfering RNAs to spectrin and siALDH2, and also treated these with HNE and TGF-beta. And Alice Chang did this analysis, and she found that if you look at the third lane, there's a profound and significant difference in TGF-beta signaling if you look at the TGF-beta members in these cells without the spectrin and aldehyde dehydrogenase 2. And it's as though the whole pathway is suddenly aberrant, and it's pro-fibrogenic and pro-oncogenic. So there's a shift. And perhaps these cleaved fragments in us, as we have a high-fat diet or drink a lot of alcohol, are doing this and disrupting normal TGF-beta signaling as we know it. Could we take this further? Alice did this through a heat map, transcriptomic analysis, and as you can see in the knockout mice, aldehyde dehydrogenase 2 and the ASCO mice, there's a marked alteration in TGF-beta signaling with an increase in CDK4 and other pro-oncogenic molecules and pro-fibrogenic. You get rid of the spectrin, you block the spectrin, and you restore everything. So this has been quite a therapeutic surprise to us. What about SMAD3 signaling? Well, if you take these cells, if you look at immunohistochemical and confocal labeling in control cells, you add TGF-beta, you'll see both spectrin and SMAD3 co-localized to the nucleus at between 30 minutes and 1 hour, transmitting normal TGF-beta signaling. You add hydroxynonenal, this reactive aldehyde, and this sequestration at the membrane. It's no longer, SMAD3 is no longer functional. You block aldehyde dehydrogenase 2, as 500 million people already have to an extent, and you add TGF-beta, and you see some SMAD3 localization, but it's no longer robust. You add H&E and you get back to membrane localization. What happens when you block the spectrum? Interestingly enough, if you block this spectrum, you add H&E and TGF-beta, SMAD3 nuclear labeling is restored. Of course, you don't see any spectrum because it's no longer expressed, suggesting that somehow if you are able to block these cleavage products, you can now restore normal TGF-beta signaling. But what about the immune cells that are regulated by TGF-beta? We performed immunohistochemical labeling and with F480, CD11b, CD4, and FOXP3 cells. And if you look in the middle bars, the ASCO mice have steatosis, fatty liver disease, and they really do look different from the wild type and the beta spectrum liver-specific knockout. And they have this marked accumulation of these purple cells. These are monocytes. There's an active monocyte infiltration, perhaps contributing to the MASH that we see in the ASCO mice and the hepatocellular cancers. And these myeloid cells have been shown by Giorgio Trincieri and Hal Moses to be very pro-tumorigenic. And it could be that these monocytes generate the tumors that we go on to see. That when you block the spectrum, now we've had these mice for a long time where we block the spectrum, TGF-beta signaling is restored, there's no monocyte infiltration, and life carries on. So targeting it with an siRNA, and now we are carrying out this work with antisense oligos. If you have 10-month mice, aldehyde dehydrogenase 2, as well as ASCO mice, and they have obesity and NASH, as you can see over here. And if you look at the far right panel, you see the ASCO mice having NASH and a phenotype of steatosis and fibrosis. You block the spectrum with siRNA for 7 weeks with 4 injections. And at that time, 7 weeks later, you then harvest the cells, which Max did, and you found that, lo and behold, the phenotype is restored to nearly normal. What about the liver-specific knockouts? The same thing happens with the liver-specific knockout. So, and on an albumin promoter, blocking the spectrum really blocks disease and prevents cleavage, we believe. Now, these mice have a systemic disorder. They are obese. And what happens to the gut, you'll ask me? And what happens, after all, it's what we're eating. And what about this male-female difference? And could we explore that further? And here we did. And if you look at the far right of the first left panel, and these are immunohistochemical labeling of liver and intestinal tissues. If you look at the far right at the bottom, there's a polymorph infiltrate in the lamina propria of the ASCO mice, not seen in the other mice. And we wondered about leaky gut. And lo and behold, these mice seen at the bottom left actually have increased fecal albumin. Clearly, we need to examine this further. Well, with Keith Crandall at GW, we wondered about this microbiome, and we're really exploring this further because it's been quite exciting. We identified that Bacteroides and Firmicutes, the pro-inflammatory members, were really increased in the ASCO mice seen in these red bars over here in the top and the bottom panels. But when we started looking at sex differences, the males have more of the pro-inflammatory species, and the females have more of the anti-inflammatory species, which really surprised us. And this could be one of the reasons that we are seeing the sex differences in MASH in our mice and perhaps in us humans. So in this part, what I've really shown you is that these ASCO mice, the spectrum has uncovered a metabolic syndrome and MASH and hepatocellular cancers in males and females, and could represent a very powerful new useful model for studying MASH and further. The mechanism seems to be these toxic adducts that you see over here in the middle panel. And the spectrum diverts from normal TGF-beta because it's no longer with SMAD3, and really it generates a competition with SMAD3, leading to fibrosis, MASH, and hepatocellular cancer, because raised levels of TGF-beta then start in a tumor-promoting manner. So targeting the spectrum, surprisingly, restored normal SMAD3 function, and possibly SMAD3 works as a partner with SMAD2, as it does in 40% of the cases, and signaling is restored. These cleaved fragments are no longer available in the nucleus and forming adducts and jeopardizing the integrity of TGF-beta signaling. So we believe that this is another approach, a non-toxic approach towards treating MASH and blocking hepatocellular cancer. And these studies could provide new insight into sex-based differences. So to continue on, we really wondered, could we harness what we were seeing in our animal models to human diseases? And could we take these tissue biomarker panels? And at this point, I'm looking at Xinwei Wang, who's done some amazing work in this field. And this is just our small snapshot where we would like to look at patients at risk and look at risk stratification and really identify tissue and blood-based biomarkers so that we can really target intervention. So, and of course, early detection using non-invasive analyses and tests would be the way to go. As you know, the major holdup current MASH clinical trials are non-invasive tests. We have to biopsy these patients, they're variable, and a host of problems, and the treatment strategies are toxic. So what is the current clinical practice? Well, current clinical practice is biannual sono and alpha-fetoprotein in cirrhotic patients from multiple different etiologies. However, very low sensitivity, and the sensitivity is reduced to even lower in obese patients. And in a third of the patients, HCC patients, alpha-fetoprotein is not raised, suggesting we really need better markers. And definitely, Xin has some amazing approaches and has identified new biomarkers, but ours is a TGF-beta-centric approach. So we wanted to look at blood and serum, query the TGF-beta pathway, and then risk stratify cirrhotic patients. How are you going to do this in sera and small samples, less than 100 microliters of sera? For this, we partnered with SomaLogic, and SomaLogic at that time really had 5,000 aptamers that they were able to analyze in the sera, and now it's 7,000. And so we took patients, a retrospective sample, and then a prospective sample that I'd like to tell you. And our goal was to really start, there's a biology, and clearly TGF-beta is really important in the biology of hepatocellular cancer. Could we take those mouse models to human disease? So these are the patient characteristics. We had 108 hepatocellular cancer patients and 359 cirrhotic patients without cancer. A total of 467, and that's grown. And this was a study which was thankfully funded by the NCI. It was a UO1 that we had. And these are the demographics, and it was done with an amazing and wonderful collaboration. It was a multi-institutional study. We were all part of this UO1 consortium, which included University of Hawaii, University of Maryland, while I was at George Washington University, and now Northwell, and we also included a few patients from UCLA. And what we identified, here's the demographics, the patient characteristics. There's very little difference in age and also the difference in ethnicity. We had a few Pacific Islanders. I don't know that we had sufficient numbers to really query sort of ethnicity and race. But these tissues and patients and patient samples blood samples and sera included hepatitis B, C, NAFLD, fatty liver disease, and alcoholic liver disease. And I'm just going to focus on one pathway. And so Alice Shang in my lab queried the first sample set that we did. Now, this is only our 55 microliters of sera that we collected, and this was a retrospective cohort of 170 patients. And you look at the bottom panel. So if you look at cirrhotic patients, just plain cirrhosis, they have a specific TGF-beta signature with genes which seem to be expressed to a lesser extent. However, when you go to cancer, there's an overexpression seen as red in these cancers. This is a left-hand panel. Now, in the middle, this black bar, there are 20 patients. These are cirrhotic patients that have a hepatocellular cancer patients signature, but they don't have hepatocellular cancer. And the question is, do these patients then develop hepatocellular cancer? And does this stratify risk? Well, this is a retrospective and a sample set, and Kirti Shetty and Ruth He and a number of people who contributed couldn't de-identify those patients. So we went on to the larger cohort, which is now 520 samples. So again, just focus on the bottom set. And there you have the cirrhotics, which really have a specific blue signature of underexpression. And the hepatocellular cancer groups, there are two, but the one to the right, group 2, has a prominent overexpression of a number of molecules regulated by TGF-beta signaling. Interestingly, in this group, we identified 119 cirrhotics that seem to overlap with both subgroup 1 and 2 hepatocellular cancers. And right now, as we speak, because we've just finished doing this on this last set, Kirti Shetty, Ruthie, and I, and Pat Latham are analyzing those, but some of those follow-ups. And I think there's a small cohort. In our case, we have about 48 patients that we followed up for over 2 years with sequential samples and studies to determine whether these people get hepatocellular cancer. So it'd be really interesting to see if we can capture that ongoing biology in the liver. But then you'll say to me, and Shin will of course say, what about these other markers? The literature is filled with a number of markers such as glypican-3 and HMGB2 and matrix metalloproteases and other molecules, not just the TGF-beta pathway. And of course, there's an enhanced liver fibrosis score, which is now going to be FDA approved for the treatment for fibrosis in cancer and diagnosing cirrhosis, perhaps, to an extent. And what are these collagens and matrix metalloproteases look like in this cohort? After all, we queried 7,000 eptimers as well as others. So Alice and Richard Amdor, our biostatistician, really collected all three and then converged to a final protein marker set and combined these with clinical variables such as alpha-fetoprotein, age, and bilirubin. And we've now come to a best final set of models for detecting, or at least even risk stratification for early hepatocellular cancer, these cirrhotic patients. And clearly this is ongoing, but this current model, which really combines what we're trying to capture in the liver, in our mouse models and human disease, to a panel reflected in the serum, could represent a promising functional biomarker panel for risk stratification. Clearly, follow-up validation studies are warranted, and we're in the middle of doing this. But to get back to my point, if you go back to these mice, they have alterations in glycolysis and gluconeogenesis and fatty acid metabolism. What about these other markers? Serine accumulates, pyruvate accumulates. ASCO mice are very abnormal, as are humans with aldehyde dehydrogenase 2 deficiency. And we are now seeing a glimpse of that. And so Adrian Craner at Cold Spring Harbor. I don't know if you're familiar with Adrian Craner's work, but he's an amazing scientist who's really looked at splicing and identified changes in splice fragments and taken this to therapeutics and neurodegenerative diseases, which are FDA approved. And he had some years ago identified that pyruvate kinase M2 has splice fragments M1 and M2, and M2 is pro-tumorigenic and involved in the Warburg effect. So we found that it was altered in our ASCO mice, and we confirmed this by immunohistochemical labeling. What about our liver-specific knockout? You can see that PKM2 is blocked, suggesting there's a macrophage. It's expressed in macrophages and Kupffer cells. The macrophage crosstalk with epithelial cells, albumin expressing epithelials, hepatocytes that can block hepatocellular cancer. And in our cohort of patients in the sera, PKM2 is increased in hepatocellular cancer as well as cirrhotics. So there's this ongoing pipeline. But what about other TGF-beta members? These mice are very, very fat, and they also have very skinny legs. So we looked at muscle mass, and together with Srinivasan Dasarathi at Cleveland Clinic, we identified that muscle mass was markedly decreased, and we examined an atypical TGF-beta, myostatin. Now, high doses and high levels of myostatin are linked to muscle atrophy, and we wondered about myostatin levels, and we found in our high-fat ASCO mice, or even on a normal chow diet, myostatin levels are markedly increased, as is increased in hepatocellular cancer. So this is an ongoing pipeline of new markers, new targeting strategies using these mouse models for human diseases. So what I've really tried to give you is a snapshot of how nearly half of human liver and GI cancers have alterations of TGF-beta members through TCGA analyses. And these mouse models could provide new insight into mechanism and blood and serum markers with targeted therapeutics to enormous value, because we're now going straight from biology and functional assays and then going back to these. A number of people have been just instrumental. Shu Rao, who's not here, she's now with the FDA, was instrumental in doing the work for the Science Translational Medicine paper and the knockout of the spectrin with Wilma, and Kazu and Kushanu have worked extensively with Paolo Cifani at Cold Spring Harbor in the mass spec studies. Alice, the bioinformatics with Richard Amdo, whom I've already told you about, and Max Yang has done all the mouse knockout work on the ASCO mice. Pat Latham and Keith Prandtl from GW have been really instrumental in our pathology as well as bioinformatics, as well as a number of people at Northwell. What I haven't told you about is a single cell analysis on the human NASH HCCs and the human MASH microfluidic cultures that we've really taken all these assays to. But most of all, I have a lot to thank NCI for the UO1 and also DCB for their funding of this work, as well as NIAAA. And thank you for inviting me. I can take questions should you wish. Thank you.
Thank you, Lopa. That was a fascinating talk. And I know, I'm sure there's going to be a lot of questions. So I know some people had to put them in the chat, but if you just want to speak up, start your camera or hide your face and speak up. Anybody has a question? So let me start with one question, which is regarding the biomarker. Obviously, I would love to see what's happening to those 112 patients that I think it was 112 that you identified with cirrhosis and that have the signature of an HCC. But what do you predict is what if you're going to treat these patients. So in other words, if they actually were to lose weight and would change their lifestyle, and we may actually have an option to change this in the near future. Do you think, based on your mouse studies, that they would reverse to the, I guess, the cirrhotic or the non-HCC phenotype?
Tim, that's a great question. You're talking about the plasticity of disease and the reversibility of disease. And for a long time, we always felt cirrhosis was irreversible, but clearly this is reversible. And diet and lifestyle regulation are very important. Having said that, there are underlying disorders. These mice, even on a normal chow diet. Now, you and I are probably forever telling our patients, whatever you do, don't eat, reduce your intake. And you're not going to gain weight unless you eat. But actually, that's not true. And there are human beings and patients who are susceptible to this. And this really unfolds the toxicity, even in a normal environment. Now, there are new studies coming out about environmental toxins for lung cancer, particularly EGFR responsive lung cancer, mutant lung cancer. I think we'll slowly find that out for HCC in this case, and really for alcohol and the types of alcohol that we drink, and also the types of fatty food that we eat, or the type of food that we eat. My mother-in-law died at 96, and she used to eat a massive amount, but she ate this very highly fermented yogurt. And clearly she had a very good microbiome. So I think as we start understanding this, we'll slowly be able to unravel this. I think we're just at the tip of the iceberg, but you raise very good points.
All right, so I see, let's start with the hand. Bingar, why don't you start, and then I ask the people from the chat. Oh, Ben, your sound is not good. Why don't you fix somehow the sound system, and then we go to Lola Reed first. I thought, right.
Hello, Lopa. Can you hear me?
Hi, good to see you.
Good to see you. It's so fascinating seeing the connections, and especially because the aldehyde, some of the aldehyde isoforms were found in incredibly high amounts in particular subpopulations of biliary tree stem cells. And so it made me wonder whether there's an overlap with what you're finding in hepatocellular carcinomas, with the cholangiocarcinomas, or in other biliary tumors like fibrolamellar.
Yeah, those are very good questions. And we haven't looked. Our mice develop HCCs, and we were surprised they don't get head and neck cancers yet. But we really do need to explore this further. They get such severe obesity. There's sort of this fatty liver disease, and then MASH has really sort of taken all our attention. But it would be important to look at these environmentally. Nowadays, we're seeing more of these mixed cancers, HCC with cholangio. And as I said to Anu, we're seeing patients at last week's tumor board. We had a patient with about 5, perhaps more, small LR3 lesions. And the question was, and a portal vein thrombus. The question was, are these tumors or not? And you can't biopsy them. And what is the reason? And why are we seeing these in 50-year-olds? And are these aldehyde accumulations generating the toxicity that our bodies are not able to take care of? Clearly, we don't know, and we do need to examine this further. Yeah, that's a great question.
All right, Ben, you want to try again?
Yeah, no, I think I fixed my the computer. Okay. First of all, Lopa, I want to thank you so much for giving this wonderful talk. And I did forward your seminar to the NIAAA. Probably some people also attend your seminar too. I have a couple of questions, for the ALG2 deficient mice. So in your hand, those mice without high fat feeding, the higher body weight, or after a high fat it's more obese, or is that
Yes, that sort of surprised us, Ben. And I know that you've really been working on the knock-in mice, and they are interesting, and they do have a very profound phenotype in themselves. But the knockouts, which we felt really gave a systemic effect, these mice are obese, and they have a significant obesity. Now, if you block the spectrum, you don't reduce the overall weight, you do reduce the liver weight, and you do reduce the fat, and it goes to this, what we call a bright fat, that brownish amber-colored fat that we see in the liver-specific knockout. Is an interesting transition. And of course, TGF-beta has been implicated in that bright fat. So clearly need to explore this further. It surprised us that we saw this phenotype on a normal diet. It's highly exacerbated on a high-fat diet, and that's when we start seeing the cancers.
But I don't think we observe. There's a paper published, in the human, especially in the Asian countries, ALT deficiency associated with obesity. I don't think that's a good report. So, the mice may be a little different from the human, or?
Yeah, it's ALDH2. If you look at some of the SNPs, they have been associated with diabetes and fatty liver disease, but it's not as frequent as the spectrum. And the spectrum seems to uncover it. We found far greater number of SNPs. We found 45 SNPs associated with spectrum. We only found a few aldehyde dehydrogenase, which would really support that study you're quoting. And you're right, it has to be ALDH2, loss of ALDH2 function to an extent, but there's something else has to unravel it.
Maybe it's one of the cofactor.
Yes, and cleaving spectrum generates this toxicity, and that might be doing it. And that could be an environmental toxin that activates caspase-3 in hepatocytes and cleaves the spectrum, and then switches on this pathological sort of network that we are seeing. This is a great question.
Another question. Sorry, another question. We always want to study all these, reactive aldehyde form the adducts with the proteins. So you study with the spectrum. Probably the many other proteins can react with acetaldehyde.
Yes.
We're trying to find a way, to think how to study that.
Yes, and I called you last year about that because I was really curious. David Crabb had done that initial adduct and how these adducts really, but acetaldehyde adducts have been shown to influence MAD3 signaling. So that's one JVC paper, but clearly we really need to see how these adducts in the nucleus start disrupting function. Now, some of these intronic SNPs are associated with, in the spectrum, associated with chromatin binding proteins. And it could be that we are looking at long-range enhancers. And as you know, this could be another way of looking at it. It may be that environmental toxins and epigenetic influences are going to play a prominent role in how our diseases manifest over the next 10 to 20 years. We're seeing that prominently in lung cancer. Because of my time as a chair at MD Anderson, I get referred a lot of patients with cancers. And right now I'm seeing a number of women, Asian women in the Palo Alto area with lung cancers who have EGFR mutations, and they're now being treated. And that's sort of, and they're young, in their 40s and 50s. So suggesting that there are these environmental toxins, and this paper out from Oxford suggesting environmental pollution can generate these mutations that we are seeing. So it does require another hit. You're right. Yeah.
Okay, thank you.
Okay, so there's a question from, I don't know, Nigusia. You want to ask the question yourself? In alcoholic drinks, there are many other chemicals and microorganisms in addition to alcohols. And how to single out the effect of alcohol to liver cancer compared to other constitutes of alcoholic drinks?
Oh, you're so right. And Tim, isn't that a great question? Because
And it's difficult to answer.
yeah, there's no good answer. There's this Italian study which said some alcohol is better than no alcohol. And they looked at those fibrosis and NASH patients. And I've always thought about that. But if you look in the country, is it wine or is it pure alcohol as we know it, or is it whiskey with all the oak barrel meshed in? Well, so does wine, doesn't it? So
Right.
I don't know the answer, and it'll be important to really start analyzing. Hopefully the RO1 will come through, looking at the effects of alcohol in these mice properly, and really just alcohol by itself.
Right.
Now,
So Sandy, you want to ask your question?
Sure. Thanks for the work and thanks for the talk. I was wondering to what extent you think that the main effect of the aldehydes is solely through interacting with proteins, or to what extent do you think they affect the DNA as well? So, for example, do you see distinct genomic mutational signature in your patients or mice exposed to the aldehydes? And also, have you compared with other cancers that are also show sensitivity to aldehydes to see to what extent similar proteins or similar DNA mutational signatures are seen?
Sandy, that's a good point. And aldehydes like ammonia, they're detoxified constantly. And as you know, formaldehyde does form these. It does alter genomic stability and DNA repair and cross-linking repair through the Fanconi anemia pathway and so on. I think collectively, it's been shown that aldehyde dehydrogenase 1 and 2 together really affect genomic stability and DNA cross-linking repair. But we haven't looked at those specifically. We did begin initially, and we published this in Hepatology, where we found that SMAD3 regulates FANG-D2 and FANG-M, and it may be that it was through SMAD3, but we never found a direct association. And clearly we need to look and determine how these aldehydes really modify DNA directly. Or is it really primarily due from these protein adducts that we are seeing in the nucleus that divert long-range or direct promoter signaling? We do need to look at that. We're in the process of beginning to analyze that. This is a great question. Thank you.
All right, and the final question obviously goes to my co-chair, Xin-Wei Wang.
Well, thank you. Well, I want to totally echo what Tim said and many others said. That what fantastic talk. And I want to keep a question short before I lose my voice. So this is a question about your biomarkers, serological data. It's quite fascinating, but would you speculating those signals, whether they are derived from cancer cells or derived from the microenvironment, particularly when you have you had a way to model those markers that that before after surgery, for example, whether they develop a relapse, what are the change over time?
Yeah, that it's a very good point. We're really looking at response to therapy, particularly with the checkpoint inhibitors playing such a huge role. And that's work from all of you, and Tim being a pioneer in this, and your recent work in the microenvironment showing that plays a driving role in these biomarkers and tumor development. We haven't looked at that. Tissue is difficult with HCC, as you know, Shin, and we've moved away from tissue. And the only way we are thinking of everything with serum readouts. So there are some companies that are able to take out the peripheral noise versus the central noise and really look at blood markers, but it's a very crude system. And hence, I think you really have to go to these animal models. And, you know, last week I got a survey from NCI saying, what do you think is important? What is the importance of animal models to you? And I was sort of thinking, oh my gosh, I can't believe I'm being asked this because without it, we wouldn't have an idea on these cells and on these pathways and so on. So it's a really powerful system to look at tissue and then query these. Now, looking at response to therapy, that's going to be a very difficult question. And I think only people like you who are doing these massive studies with Anu are in a position to really do those because they're really labor intensive and they require unusual and innovative bioinformatics. And similarly to Ben, with the aldehydes and these adducts and how to really address that, because I think that's centrally located at the NIH. And Sandy and Lola answer those questions with other types of cancers, which are also very prominent. But thank you very much.
Okay, Lopa, thank you again. That was a wonderful talk. Thanks everybody for an interesting discussion. And I don't want to close before telling you that we're going to have the next talk on February 26th. We're going to have Amit Singhal from UT Southwest as a speaker. And I hope I will, like many of you, will join in February and have a good rest of your day.
Yeah, and thank you. And thank you, Anu, and thank you, Cross Thompson, for really.
Thank you, Lopa. See you at 3:30.
Thank you. Yes.
Thanks, Lopa. We'll see you
Thank you.
tomorrow.
Bye-bye.
Bye.